Hey, welcome back. This is biology question 39 from IMAT 2017 past paper. So I will be answering this question. So it says, which one of the following is correct outline of some main events in photosynthesis in healthy wheat plant? Okay, so before we go through each of these options, we first must recall the basic and the most important definitions of about the photosynthesis. So photosynthesis is quite the most general term, photosynthesis. So basically it means any organism which consists of chlorophyll, chloroplast that uses it to synthesize their own food. Okay, so that's the basic term. So in a more biological detail, it's conversion. It's conversion of light energy to chemical energy. So the chemical energy for the most organisms, the photosynthetic organism is a glucose, which this would be then further broken down to be used as an ATP. Okay, so the main idea is that the light energy, which is the photon light from the sun, is being converted into chemical energy, which is glucose, and further converted into ATP. Okay, so during the respiration. Alright, so this is the main idea about the photosynthesis, that it occurs in most photosynthetic organisms like the green plants and the algae. Okay, so these are the common uh, organisms. Okay, so now photosynthesis is divided into two parts. So I'm going to mention here photosynthesis. Okay, so photosynthesis is divided into two parts. So first of all, we must know that photosynthesis primarily occurs in the chloroplast. Chloroplast, which is the organelle. Okay, so as I mentioned, so the photosynthesis is divided into two parts. So the first part is light dependent reaction. Okay, so light dependent reaction, and the other one is light independent independent reaction. Okay, so light dependent reaction and light independent reaction. So light dependent reaction primarily uses light to process their reaction, but the light independent reaction does not use the light. Okay, so the light dependent reaction uh, has one important term that defines this reaction, which is called photophos pho- phosphorylation. Okay, so photophosphorylation is the one main important process of the light dependent reaction. And for light independent reaction, the most important process is what we call as Kelvin cycle. Okay, so now the main function between these two is that the phosphorylation uses the phosphorylation has its main products that include ATP and NADPH. So these two high energy molecules, and also the other product which is considered the waste product for the plant itself, is oxygen. Okay, which is the byproduct, so it's released as a waste product from the plant. But the main product, the most useful one, are ATP and NADPH. So ATP is the main energy currency for the organism, and the NADPH is also high energy molecule, but most importantly, it is a reduced coenzyme. Okay, so these are the main products, but the reactants that are used in this process is the water. So the water is converted all into these products. And whereas for the Kelvin cycle, the since the Calvin cycle is light independent reaction, so what it does is it basically converts the reactant that we have carbon dioxide into glucose. So this is where the final process ends. So carbon dioxide into glucose. So we also must know that these processes, light dependent and light independent, are interconnected. So they're not the separate process, they're interconnected to each other. So how it is that phosphorylation main focus is to produce high energy molecules, which is ATP and NADPH. So these high energy molecules are being fed into this Calvin cycle to power up this cycle to help in conversion of carbon dioxide into glucose by using these high energy molecules ATP and NADPH. Okay, so these processes are interconnected with each other. So now I'm going to explain in further detail as to how these two processes are interconnected in sustaining the plant's life. Okay, so now uh, moving on to this diagram. So the first of all, this diagram that I'm showing here is the diagram of chloroplast. Okay, so this organelle, this chloroplast, which is also known as double membrane, double membrane bound organelle. Okay, so this organelle is double membrane bound organelle. So this is the main organelle where the photosynthesis reaction entirely occurs. Okay, so now I'm going to label the most basic parts of the chloroplast that you must know for almost every IMAT exam. Okay, so now this first outer layer of the chloroplast is outer membrane, outer membrane, and this inside membrane is the inner membrane. So these two structures together is the reason why chloroplast is known as double membrane bound organelle because it has two membrane, outer membrane and inner membrane. Okay, so now this part that you see the light green over here is known as a stroma. So stroma is basically a jelly-like fluid. So it's also aqueous fluid, jelly-like fluid matrix. So similarly as how the eukaryotic and prokaryotic cells have the jelly-like fluid which we call as cytosol or cytoplasm. So similarly, the chloroplast has its own jelly-like fluid which is called stroma. Okay, so and the next one is these things that you see, the rectangle-like thing, the disc-like structures. When you stack these discs together, these discs are called, these are stacked discs are called grana. Okay, and these individual disc-like structure are called thylakoid. Okay, so these individual disc structures are called thylakoid, which is a disc-like structure, and granum is a stacked thylakoid. So meaning if you stack all these individual thylakoids together, it forms a granum structure. Okay, so now the next one that we have here is these are stretch-like structure that are called lamella. So basically lamella is like the structure of the thylakoid is like an extension of thylakoid membrane. So basically it's to interconnect all these thylakoid membranes together. Okay, so This is the main structure, and also the most important one is that this open space is also of the thylakoid. Okay, so this part over here is a thylakoid membrane, thylakoid membrane, and the inside structure is called the thylakoid lumen. Okay, so this inside structure of the thylakoid disc is called lumen, the space inside the thylakoid. Okay, so now this is the most basic structure that you must know for IMAT exam to solve every question related to photosynthesis. Okay, so now this is the basic anatomy of the chloroplast. Okay, and this light blue-like structure that you see is just the cytoplasm of the cell. Cytoplasm, or you can say cytosol to be more clear. Cytosol. Okay, so this is the basic structure. So now moving on forward. So now the structure that I'm showing you here is the structure inside the thylakoid membrane. Okay, so wait, let me be clear. Okay, so this membrane that you see over here, the light, this green part is a thylakoid membrane. Okay, so now what this structure exactly is that as I've mentioned over here, this thylakoid membrane, the one that I've circled over here. So this diagram over here is the zoom in structure of the thylakoid membrane. So I've enlarged the picture of the thylakoid membrane to show you exactly how the structure looks like. And this part is stromas as the stroma surrounds each thylakoid, because this thylakoid outside of the thylakoid membrane is the stroma, this jelly-like fluid structure. Okay, so this is stroma, and this is thylakoid membrane, and this is the lumen, which is the inside the space inside the thylakoid structure. Okay, so now the significance of this structure is that I'm going to show that how the photosynthesis reaction occur, but more specifically, inside the thylakoid membrane, the reaction that occurs is known as light dependent reaction. Light dependent reaction. Okay, so this is where the light dependent reaction occurs in this structure, which we also call as photophosphorylation. Okay, so photophosphorylation. So why we call photophosphorylation? Because it uses the word photo means use of light energy, and phosphorylation is to phosphorylate, meaning the production of ATP using ADP. So ADP and pi are connected to each other to form ATP by use of light energy. So now I'm going to explain exactly how this process is work. So now we have these integral membrane proteins over here that you can see. So this first structure that you see is PS2, which is the full form is photosystem 2. So what the structure does is the most important thing about it is this is the structure where the chlorophyll is located. Okay, chlorophyll, the green pigment of the plant. So this is exactly where the chlorophyll is located. So the function of this structure is that it absorbs the photons of the light. Okay, photons, or you can say the light rays. Okay, so it absorbs the photon using the chlorophyll structure. So as the photon is absorbed, it contains an electron. So this electron is being excited to higher energy level by use of photon. So once the electron reaches its high energy state from the photosystem 2, the high energized electron moves across a series of proteins that are found in this thylakoid membrane. Okay, so there are series of very small proteins that are located in the thylakoid membrane. So it moves through these series of proteins in the membrane or the thylakoid membrane, and it reaches to this proton pump channel. Okay, so the function of the electron in this proton pump channel is that it provides the energy for the this pump channel to pump the protons from the stroma into the lumen. So most of the time, the proton pump is an active process. So it has to pump these protons against the concentration gradient because there are more amount of protons that need to be pumped inside the lumen. Okay, so it has to be an active process to pump against the concentration gradient. So the energy provided for this process is by this high energized electron that is provided by the photosystem 2. Okay, so now once it reaches to this destination, the high energized electron does not stay here. It again moves into series of the proteins, the small proteins that are found within the thylakoid membrane. So it reaches to photosystem 1. So what it does here is the exact same process happens as in photosystem 2. So the main function of this is the same thing as photosystem 2. It uses the photons of the light to re-energize the electron to higher energy level. So now what happens is now once the electron in the photosystem 1 has been re-energized to higher energy state, the high energy electron will now move across the series of protein, and it will reach to its final destination, which is uh, NADP plus reductase. So now this is a peripheral protein structure. So NADP plus reductase. So now what the function of this NADP plus reductase is that the NADP plus reductase uses this electron to pass to a structure which is NADP plus. So this compound over here gains the electron from NADP plus reductase, and the NADP plus reductase also combines the proton with the NADP plus, and along with this, the electron is also combined. Okay, so the electron is gained to form NADPH. So now this is the final structure, which is the reduced coenzyme. Okay, so now the structure that we have over here is the reduced coenzyme, the high energy molecule. So this molecule is the electron carrier. So it carries the high energy electron to its structure, which will be used in the other processes. Okay, so the NADPH is produced in the NADP plus reductase. And the most important thing about this process is that NADP plus here act as a final electron acceptor. So this is the very most important step of the final part of electron is that here NADP plus accepts the electron, accepts as a final uh, electron acceptor from this reductase structure and combines with the proton to form NADPH. Okay, so this is the final part where the electron is gone, but it is recycled in the most useful way. Okay, so now what happens here is that the process does not stop here. Now we know the electron from the photosystem 2 has been lost, as it has been used up in the end by the NADP plus to be converted into NADPH. So now to replace this lost electron, what happens is the photosystem 2 uh, utilizes the photon to carry out the most important reaction, which we call as photolysis. So the water over here, which is the main reactant, is converted into, it is splitted into oxygen and two electrons and four protons. So I'm going to have to balance the equation. So this is two water molecule producing these co- producing these products. Okay, so two water molecule produces these products. So this process is known as the photolysis. Photolysis. Okay, so this process is known as photolysis, where the water molecule is converted, is splitted into oxygen, two electrons, and four protons. And this reaction primarily occurs in the photosystem too. Okay, so here these electrons are now going to replace the last electron from the photosystem too, so that the reaction uh, keeps continuing, so that the reaction keeps continuing and to produce the essential compounds, which are the reduced coenzyme and the ATP. Okay, so now I'm going to explain how the ATP is produced from this process. Okay, so as I've said, the proton pumps. So we have the most important structure over here, which is called ATP synthase. So this structure, which converts ADP into ATP. Okay, so now how it does is that the proton pumps that you see over here, as I've explained before, it pumps the proton against the concentration gradient. So inside the lumen of the thylakoid membrane, we're going to have a lots of protons over here. So now what happens is that once the proton reaches its suitable concentration gradient, so the proton is going to move down its concentration gradient through ATP synthase. So like that, it's going to move out. So now this process where the proton moves down the concentration gradient through the ATP powers up the ATP synthase. So ATP synthase is powered up. So the best analogy I can give you is like ATP synthase, like you can imagine this as a turbine. In a turbine in a dam where the huge amount of water powers up the turbine and converts into electricity. So similarly, these protons are like a water in the dam. So once we have high amount of protons in the lumen, these naturally move down their concentration gradient through this turbine-like structure, ATP synthase, to power this up. So once the ATP synthase is powered up, it converts ADP plus pi into ATP. So this is where the ATP is generated by use of high concentration of protons moving through ATP synthase and out of the lumen and into the stroma. So this process powers up to result in ATP synthase to convert ADP into ATP. And the most important term that we define this, where the ions that moves across the ATP synthase to power up to synthesize ATP by the concentration gradient. This process is known as chemiosmosis. Chemiosmosis. Oh, sorry for my spelling. I hope this is correct. Okay, wait. Chemiosmosis. Okay, so this process where the ions naturally flow down their concentration gradient, which results in the synthesis of ATP due to the concentration gradient, is called chemiosmosis. Okay, so this process is chemiosmosis and it powers up the ATP synthesis to produce ATP. Okay, so now this is the entire structure of the photophosphorylation, where now we have the most important compound and product that are produced is the NADPH, our reduced coenzyme, and ATP. So these two high energy molecules are now going to be fed into this structure. So this one is this clear structure that I'm showing you here is the stroma. Okay, so this is the stroma, which I've explained is the jelly-like structure. Okay, so this stroma is the place where light independent independent reaction occurs. Independent reaction occurs. Okay, so which we also call as Kelvin cycle. Okay, so light independent reaction, which we also call as Kelvin cycle, occurs in the stroma of the chloroplast. So these two structure I'm showing you here is just the structure inside the chloroplast. Okay, so in the entire photosynthesis, as I've explained, photosynthesis entirely occurs inside the chloroplast. Okay, so now this structure primarily occurs in the lumen of the thylakoid. Now this is outside of the thylakoid membrane, which is the stroma. Okay, so this is where the Kelvin cycle reaction occurs. Okay, so the first of all, uh, the main compound, the main reactant that are used in the light independent reaction is carbon dioxide, which I'm going to mention as 1C, which means one carbon compound. Okay, so now you'll understand later as to why I'm explicitly mentioning that this compound is one carbon compound. Okay, so now the other structure the other compound, the most important one that we have over here is RuBP, which is a five carbon compound. So this compound is known as acceptor compound. Acceptor compound. Okay, so now what happens over here is that these two compounds combine together to form a structure to form a compound, two molecules of GP, which is a three carbon compound. So each GP molecules are three carbon compounds. So two molecules of three carbon compounds are produced, which is a GP. So this reaction is most importantly catalyzed. So this reaction is catalyzed by enzyme called Rubisco. So this is an enzyme that catalyzes this reaction. And this reaction, the most important name for this reaction is known as carbon fixation. Carbon fixation. Okay, so this process where the carbon dioxide RuBP are combined together uh, to form two molecules of GP, where the reaction is essentially catalyzed by this Rubisco enzyme. This entire process is called carbon fixation because the carbon dioxide is being fixated into RuBP to form this new compound, the two molecules of GP. Okay, now these two molecules of GP is going to be converted into two molecules of GALP. Okay, so these are also a three carbon compound structure. Okay, and this process requires the high energy molecules that I've mentioned in before, which is 2 ATP and 2 NADPH. Okay, so these high energy molecule is where it's most important. Okay, so now this entire uh, this process or this step is known as reduction because the GP molecules over here, the two molecules of GP are reduced into two molecules of GALP. So this reduction essentially occurs by this NADPH, which provides the proton to this two molecules of GP to be reduced into GALP. Okay, and this process is requires energy, which is provided by ATP. Okay, so the ATP provides the energy for this process to occur. Okay, so now this is the reduction stage. So now this is where you have to analyze why the light independent reaction, the Kelvin cycle, are interconnected with light dependent, the photophosphorylation. So as I've mentioned, uh, the main products from the photophosphorylation is NADPH and ATP. So these two high energy molecule is where used in this reaction. As I mentioned, the two molecules of GP is converted into two molecules of GAL. So ATP and NADPH is used in this stage. So that's why they're highly interconnected with each other. Okay, so now what happens is that the reaction does not stop here because this reaction is a cycle. So it loops back to continue this process. So how it does that, the GAL is going to be converted back into RuBP, and but there's a catch. It's not entirely going to be converted into RuBP because it has to split into two parts. So what we know is that the two molecules of GALP, which is a three carbon compound, has a total net of six carbon. So this is total net of carbon. So the total carbon that we have is six carbon because two multiplied by three carbon, we have six carbons total. So from this six carbon, one carbon is going to be kept safe, which is acting as a reserve for glucose. Okay, so reserve for glucose. So this one carbon is going to be kept as a reserve for glucose, which is used to synthesize glucose. And the other remaining five carbon from this carbon, uh, six carbons is going to be used to regenerate RuBP. So this regeneration of RuBP also essentially requires ATP to power this process up. ADP plus pi. Okay, so this also requires an ATP, and this step of converting the GALP into RuBP is known as RuBP regeneration. Okay, so RuBP regeneration. So this requires an ATP to convert this GALP into RuBP, but not in the entire GALP is converted into RuBP because we know that this entire part has total of six carbon. So the six carbon is split it into two. So one carbon from six carbon total net is reserved for glucose, and the other remaining five carbon are being used to regenerate RuBP, which is followed by the ATP to power this process up. Okay, so now since it is just a one carbon, the cycle is going to repeat six times. Okay, so it has to occur over six times till it finally reaches six carbon reserve for glucose, where it can finally have produced completely produce glucose completely, because glucose is the formula for glucose, it's a 6 C6H12O6, so it requires a six carbon, so it's just the one carbon reserve. So this cycle has to repeat over six times so that it produces one glucose molecule from this six cycle completely. Okay, so now this is the entire process of the Kelvin cycle, which occurs in the stroma of the chloroplast. So what was the main idea about this entire diagram that I'm showing you here is that how the light dependent reaction and light independent reaction are interconnected, because the light dependent reaction primarily produces high energy molecules NADPH and ATP, which are being fed into this cycle, Kelvin cycle, to power up this process, where ATP is being used over here and NADPH, which is to reduce this compound GP into GAL. Okay, so now that's the entire diagram of the photosynthesis. So these are two reactions together that makes up the photosynthesis. Okay, so now we can go back to our question and solve it. So since these are the most important thing that you must know for this question. Okay, so now going back, so now we can read each options to find out which are correct about the process of photosynthesis. Okay, so the option A says carbon dioxide combines with acceptor compound which breaks into two, and each is reduced by hydrogen split from water by light. Okay, so now this is easily, we can mark it as correct as I've explained the entire process over here. Okay, so the simple thing is that it mentions carbon dioxide combines with an acceptor compound. So as I've mentioned, the acceptor compound over here, which acceptor compound, which is RuBP. So which is the 5 carbon compound. So the acceptor compound that they're referring to is RuBP. And this entire process where the carbon combines with acceptor compound to produce the into two. So this into two is GP. The two molecules of GP, which is a 3 carbon compounds. And so this entire process, carbon dioxide combining with the RBP to form two molecules of GP is known as carbon fixation, which I've explained in the Kelvin cycle. Okay, so this process is still a Kelvin cycle, and each is reduced by hydrogen. So the ones that are each are reduced by hydrogens are the two molecules of GP, and when they're reduced, they produce gal. Okay, so they're reduced by hydrogen. So the one that carries the hydrogen for this process is the NADPH. So this structure is the most important one that provides the hydrogen for this reduction reaction to reduce GP into gal. Okay, so reduced by hydrogen. So this process is done by NADPH. So NADPH acts as reducing agent over here. Okay, so reduced by hydrogen split from water by light. So this part is explaining about photolysis, which I've explained in the photolysis, which I've explained before in the light dependent reaction, where the photolysis occurs in the photosystem 2, where the water molecule is split into oxygen, 2 electrons, and 4 protons. So these protons are provided by the water molecule that are split using a photolysis process in the photosystem 2. Okay, so that's what they're referring to, split from water by light. So that's why this option is correct. Okay, so now we can go through other each option and find out why other options are wrong. Okay, the light option B states light splits water and resulting combines hydroxyl group combines with a compound which has reacted with carbon dioxide. So we can easily mark this as irrelevant because light does not split into hydroxyl. Light splits the water molecule, which this process is explaining about photolysis. So as I've explained before, the balance equation, the water molecule in the photolysis is splitted into oxygen and 2 electrons, and we have here is 4 protons. Sorry, it's not. Okay, 4 protons. So as I mentioned over here before, two water molecules into these products, which is photolysis in the photosystem too. So this statement explains about the photolysis, but it's wrong because it mentions hydroxyl group. We don't have hydroxyl group. We have oxygen, 2 electrons, and 4 protons as our main product in the photolysis. Okay, so now the option C. In the presence of light, oxygen reacts with carbohydrate to produce water and carbon dioxide. And this one is also wrong because why? In the presence of light, oxygen reacts with carbohydrate. That's completely irrelevant. In the presence of light, this statement is trying to explain about the light dependent reaction, which is a photophosphorylation. So we do not have oxygen as our reaction. Oxygen is considered as a product of the light dependent reaction. It does not react with any it does not react at all. It is just disposed as a waste product from the plant. That's all it happens. So it's a product, it's not the reactant, so it does not react. Okay, so you can easily pinpoint out this as wrong. And option D, the light splits carbon dioxide and resulting carbon then combines with oxygen and hydrogen obtained from water. So this is completely irrelevant. Light does not split carbon dioxide. Light is used in photolysis to split the water molecules into oxygen and these products that I've mentioned here. Option E states light joins carbon dioxide to an acceptor compound. So, okay, acceptor compound which is then reduced by hydrogen obtained from water. Okay, so now this option quite looks similar to as option A, which is quite tricky. So now you have to be careful. So light joins carbon dioxide. So you can easily say this is wrong. Light does not join carbon dioxide. Carbon dioxide is used in the Kelvin cycle, which is light independent reaction. So light occurs in the light dependent. So carbon dioxide does not is not involved in the light dependent reaction. Okay, so you can easily cancel this out. So option E is wrong. So that's why option A is correct. All right, so that's all about this question. So thank you guys for watching my video. I hope you find this helpful. Please leave a like and subscribe to my channel. Thank you guys. Bye.